And we said that there were basically two main types that we spoke about, electrical. Yeah. All right, and chemical. And we said some basic properties or descriptions of electrical synapses, right? We said they weren't that important, but we did distinguish them in some properties. And does anyone remember what those were? So what's something that sets an electrical impulse apart from a chemical one? They're bidirectional, perfect. So they're bidirectional, meaning they facilitate the flow of this impulse in either way, right, from cell to cell. And in order to be bidirectional, what structure did we mention facilitates this bidirectional movement? Gap junctions, perfect. So they require gap junctions, which are the pores or the passageways that we talked about, which created that open flow of communication. What else did we say was different about the type of transmission, the speed? Really fast, so much faster transmission. Okay, and then lastly, we said that they are aware and not well understood. Okay, all right, so that's electrical impulses, and this is literally all I expect you to know about electrical impulses. Chemical impulses. What properties did we say about these? So in contrast to being bidirectional, chemical impulses are unidirectional, right? And we looked extensively at why, the nature of the action potential, and that saltatory conduction makes these unidirectional. And again, in contrasting, what about the speed of these? They're going to be slower, right? So they're going to be slower transmission. And can anyone remember what element of this made this a little bit slower? We talked about a specific reason that this was sort of delayed or halted or a little bit slower than electrical synapses. All right, so we talked about the synaptic delay, all right? And so they're a little bit slower because of that synaptic delay. All right, can anyone remember the cause of the synaptic delay? So we said it was not one factor and it was something else. And does anyone remember those two things we talked about? The cause of that 0.5 to 5 millisecond delay. Am I seeing a hand? Perfect. So time for the vesicles to dock, to fuse their membrane with the membrane of the axon terminal, and to exocytose that neurotransmitter. So this was really due to that exocytosis process. Okay, make sure you review that. It was not due to the diffusion. It was not due to any time of the neurotransmitter to move across the cleft, but rather the time it took to dump that neurotransmitter out. Okay, very important distinction. We also said it required a few players, chemical synapses. Does anyone remember the two players we talked about? We sort of introduced them last time. So there are structures that are required for a chemical synapse, but not for an electrical synapse. We talked about neurotransmitter. Right, neurotransmitters are required here. And again, just to sort of distinguish here, neurotransmitters can be a lot of different things. We sort of touched on this last time. They can be proteins, amino acids, they can be peptides, amines. And so it's not so much of a structural differentiation here, but a functional one. And the job of a neurotransmitter is simply to transmit that signal. Okay, it's going to be stored in a vesicle on one neuron. It's going to cross the synaptic cleft, bind to a receptor on the other neuron, and as such, it's simply transmitting the signal. Okay, it's a chemical used to transmit that signal. And so because there are lots of different neurotransmitters at lots of different locations across the body, likewise, we have lots of different receptors, right? And these are sort of the two players that are required for this chemical synapse. We need neurotransmitter, we need receptors. One of the most common neurotransmitters we'll talk about is acetylcholine. We'll talk extensively about that. It's the most abundant neurotransmitter in the body. And it's going to bind to a very special type of receptor. It's called a cholinergic receptor. So for every neurotransmitter that's sort of unique and different in function and location, there's likewise a unique receptor, okay? We're going to look at different categories of these neurotransmitters and receptors. But we spoke about two broad categories of the receptors. And this was based upon their response or their speed on the postsynaptic membrane. Can anyone remember the two broad categories? Or the two broad types of receptors? Okay, so we talked about ionotropic receptors. And we talked about metabotropic receptors. Okay, and which of these were fast and which was slow? All right, so we said ionotropic was faster. Okay, and that was because the receptor is the channel, right? The receptor is acting as a channel. And so when a ligand, which in this case is our neurotransmitter, binds to that receptor, the receptor and the channel are one, there's no middleman in between, and so we immediately bring about that cellular response. So very quick process, very little steps or players involved, and so that becomes very quick, very fast. Metabotropic, on the other hand, are slower, and that's because there are a lot of players involved, okay? And that's sort of the best way to compare this. There are lots of different steps in this sequence of events, and as a result, we call this the signal transduction cascade. And when we say signal transduction, just equate that word to mean that there's a passing of the message from different players, okay? That signal or that message is being passed along, okay? We'll talk about the players that are involved there. So it's slower in its effect and its response on the postsynaptic cell, and that's because we have a G protein, right? It's coupled to a G protein that's going to do something else in that cell. It can do one of two things. It can be directly coupled, and I'll illustrate what these mean again. I'm just writing them here. We'll review this shortly. So this G protein could do one of two things in the cell. It could be coupled to some sort of G protein. It could be directly coupled to an ion channel, right? A part of that protein can activate this ion channel, or it could synthesize some second messenger. All right, so not to get confused here. Metabotropic receptors are slower, they require a G protein, and then that G protein could do one of two things. It could directly be coupled to another ion channel, or it could secrete some second messenger, which we'll talk about, that will then activate a cellular response in the cell.